Now, today I shall discuss the anatomy of the inner ear. Though it is very complex to understand, because it is located deeply inside the temporal bone, and micro dissection or dissection is not shown everywhere, and you cannot get the proper view of any living or any until you see the operation. The anatomy of the inner ear, so I discuss in a very easy manner with diagram in a PowerPoint presentation. So the component of the middle ear, the inner ear, are two. That is, it is a bony component, and inside the bony component there is membranous labyrinth. The bony labyrinth, actually, it is a periosteal covering of a space, and inside which this space there is a membranous labyrinth. So there is bony hollow. We can divide this bony labyrinth into different parts. That is bony vestibule, where the membranous vestibules is located there, where the sacules and utricle, the bony cochlea, and the semicircular canals, bony. The bony labyrinth houses the membranous labyrinth, that is the inner ear or labyrinth of the ear, also called the labyrinth of the ear. It can be divided functionally into two parts. That means ear, middle ear, inner ear, that is both balancing function as well as the hearing function. So the nerve is called the vestibulocochlear nerve. The vestibule is concerned with the balance, that is the vestibular system, and the anterior and medial part is cochlea, that is which laws the membranous duct is mainly for the hearing. The vestibule consists of utricle, sacules, and the semicircular canals, which are inside the bony vestibules. Similarly, the membranous semicircular duct, which is housed inside the bony cochlear duct, consists of the scala media, the scala tympani above, and scala vestibuli. The scala vestibuli above and the scala vestibuli below. Now, let's go to this. The component are bony vestibules. There is inner endochondrial layer and the outer periosteal layer, and intermediate in between the bone there is endochondrial bone. So the thickness is there. It is not just seat. The thickness in the outer aspect is harder. The bony vestibule is rigid, and the bony within a rigid, and it is within the temporal bone. The vestibule is the hardest part of the body other than the enamel. It consists of three parts: the vestibule, bony vestibule, the bony semicircular canal, and the cochlea. The cavities is hollowed out in the substance of this bone, lined by the pelvis. It contains a clear fluid, that is sorry for the interruption. Sorry for the interruption. The central part, the vestibule is the central part of the bony labyrinth, is separated from the middle ear by the oval window and communicates anteriorly with the cochlea, posteriorly with the semicircular canal. The hollow bony cavity, it lodges the membranous labyrinth. So inside it, the bony vestibule lies the membranous labyrinth, consists of saccules and utricles. On the lateral wall of the bony vestibules, there is a bean-shaped opening covered by the footplate of the stapes. On the lateral aspect of the bony vestibule is a bean-shaped bony covering covered by the footplate of the stapes, surrounded by the annular ligament, that is, which is called the. This is the picture of the bony vestibule. The outer aspect is the oval window covered by the annular ligament. This anteriorly there is cochlea and the posteriorly there are semicircular canals. The superior semicircular canal, the lateral semicircular canal, and the posterior semicircular. These are bony. This bony semicircular canal, both have one is the ampullary end, that is swollen portion, like a, and another is tubular portion. Here also the superior posterior semicircular and ampullary end. The posterior semicircular has another tubular end. Both the tubular end, the superior and the posterior semicircular canal, joined together to form a common opening, a common canal called the cast communis. But the lateral semicircular canals opens inside the bony vestibule as separately in the two openings. Now, inside the bony opening, in the front part of the medial wall of the bony vestibule is a spherical depression called the spherical recess. So, anterior part of the bony vestibule is a spherical recess. That is a depression or a area, which this area lodges the saccule. The macula of the tubular media, is perforated by means of minute holes from the passage of the inferior vestibular and filaments. The posterior superior portion of the medial wall of the bony vestibules, there is another depression, or in the medial wall we can see another depression called elliptical recess. This area lodges the utricle. Between these two recesses, that is a spherical recess anteriorly and the posteriorly the elliptical recess, there is a ridge, a slight elevation called the vestibular crest, and anterior end being the vestibular pyramids. The vestibular pyramids is a part within the thing. The elliptical recess and the pyramid will have minute perforation or foramen-like structure called the macula tubulosa superioris, or also called the myclots, for the passage of the filaments of the nerve from the utricle and the ampulla of the superior semicircular canal, the lateral canals, and the inferiorly and the vestibular crest splits to enclose the cochlear recess. The inferiorly, the vestibular crest splits to enclose cochlear recess for the cochlear nerve. Below the elliptical recess, there is another opening, that is the aqueduct of the vestibule for passage of the endolymphatic duct. In the posterior part of the vestibule, there are 5 openings again total. We see two opening for the lateral semicircular canal, 2 for the ampullary end of the superior and posterior semicircular canal, and 1 for the cast communis. So there are 5 openings. You can see this is an opening, this is another opening, this is another opening, the common class. So there are lateral semicircular canal. There are 5 openings, and the depression of, after removing the membranous semicircular canal, membranous labyrinth, we can see the spherical recess anteriorly, which for the saccule with the vertical crest and the pyramid. This fits the lower end of the vertical crest fits to enclose a aqueduct of the cochlea. And the semicircular canals are the bony semicircular. There are three bony circular canals on each side, occupying 2/3 of a circle, having a diameter of 0.8 millimeter. Superior, lateral, and posterior or vertical. These are orthogonically related over an arc of 240 degrees. These angles are formed by the semicircular canal. It is known as the solid angle. That is, solid angle is actually not an angle. It is a core of bone between the three semicircular canals. So between the angles of the semicircular canal, the area, this is the angles of the semicircular canal. This is the semicircular canal. So this is the solid angle, the core of bone, which is in between the three semicircular canals. Now, another point is Todman's triangle. The Todman's triangle is a triangular zone bounded anteriorly by the bony labyrinth, the posteriorly by the sigmoid sinus, and superiorly by the superior pectoral sinus. That is, through this Todman's triangle, the infection can go to the posterior portion, leading to cerebral abscess and complications. In all the semicircular canals, has a one ambulatory dilated end called the ampulla, and another is non-ambulatory. So we see, because the non-ambulatory end of the superior and the posterior joins together forms a class combination. So we have 5 openings for the 3 semicircular canal. Now you see, these are the semicircular canals, bony semicircular canals, lateral, inferior, posterior. So now about the cochlea. The cochlea resembles like a snail. It is coiled tube, bony tube coiled, making 2.5 or 2 3/4 turn around a solid core of bone, which is called the modulus. The cochlea houses the membranous cochlear labyrinth or membranous cochlear duct, which is the ordinary part of the inner ear. It twists on itself over a central portion of the bone called the modulus, producing a cone-shaped and pointed towards the anterolateral direction. The branches from the cochlear portion of the vestibular nerve are found at the base of the modulus in a spirally arranged manner with multiple foramens called the tractus spiralis foramen. So it is a conical structure. The anterior end of the cone extending outward from the modulus. Extending outward from the modulus is a wedge of bone that is from the modulus. The winding roundward, around it, there is bone. So this bony projection, self-like projection from the modulus in the bony cochlear duct called the spiral canal. That is osseous spiral lamina. These osseous spiral lamina, along with the is incomplete. It is completed by another. So two membranous like structure. One is the basilar membrane and another residual membrane. So the scala vestibuli is located superiorly to the cochlear duct, because cochlear duct is attached to the spiral lamina. It has upper superiorly the retinal membrane and the inferiorly the basilar membrane. So this located superiorly to the cochlear duct is the Scala vestibuli located inferiorly to the cochlear duct or below the basilar membrane. It is the scala tympani. The scala vestibuli ends continuous to the scala vestibuli suggests it is continuous to the vestibule, and whereas the scala tympani it terminates to the round area. The bony cochlea spiral to an bony cone of bone. The base of the modulus is directed toward the fundus of the internal auditory meatus and transmit vessels and nerves to the cochlea. The apex is towards the anteriorly, almost towards the tensor tympani muscle. It is the modulus spirally arranged scleroid, that is osseous spiral lamina, and its incomplete division is completed by the attachment of the basilar membrane from the free edge to the osseous spiral lamina, that is outer aspect of the bony canal. Similarly, another is called the basilar membrane. Another membranous structure is a basilar membrane. The membranous labyrinth, within the bony labyrinth is the membranous labyrinth. Membranous labyrinth consists of the cochlear duct and three semicircular canal, the utricular saccules, and the endolymphatic duct and sac. The cochlear duct is endolymph filled fluid inside the cochlea. The cochlear duct is an endolymph fluid. You can see, triangular in cross section. This is the osseous spiral lamina. This is osseous spiral lamina. This is the retinas membrane. So this is the cochlear duct, membranous cochlear duct. The upper portion is the scala vestibuli, lower portion is the in the diagram is the scala tympani. So the cochlear duct is triangular in cross section. Is triangular in cross section at the middle portion of the bony cochlea, attached to the free end of the spiral lamina, and laterally to the stria vascularis. Laterally to the stria vascularis, spiral lamina and stria vascularis. The floor is formed by the basilar membrane. The roof is by the vesicular. The floor is formed by the basilar membrane, and roof is by the retinas membrane. The content and communication. The cochlear duct is located within the scaffolding of the bony cochlear duct. It is held in position by the spiral lamina. And the presence of this duct creates two channels. Above is the scala tympani and below is the scala tympani. Above is above and below. Above is the scala vestibuli and below is the scala tympani, respectively. The cochlear duct or the scala media ends a little ahead of the apex. Thus, the gap remains persist. So, the gap is called the helicotenum, through which the scala vestibuli and the scala tympani communicate freely. Both the scala tympani and the scala vestibuli contain perilymph. So, there is a hole. Why the hole is necessary? The hole is necessary because when there is a movement of the stapes, there is movement of the fluid inside, and the fluid waves ultimately turns above and goes. So it allows the movement of the stellar tympani, thus the movement of the basilar membrane. The communication, the scala vestibuli is closed by the foot product stapes, which separates it from the media, whereas scala tympani is closed by the secondary tympanic membrane or the round lunar membrane. The scala tympani is connected to the subarachnoid space also through the aqueduct of the cochlea. The interior of the cochlea. The cochlear duct can be described having a triangular in shape. Lateral wall formed by the thick periosteum, known as the spiral ligament. Loop is formed by the membrane which separate the cochlear duct from the pili vestibuli, known as the Reissner membrane, and the floor, the membrane which separates the cochlear duct and the scala tympani, known as the basilar membrane. The basilar membrane houses the epithelial cells of hearing and is the organ of body. The endorphic duct originates at the medial wall of the vestibules. It first parallel to the pars communis and then posterior semicircular canal and to heads to the endoventricular sacs, the anterior and the posterior, anterior medial to the sigmoid sinus. The endorheipatic sacs lies approximately 10 millimeter inferior and lateral to the boras of the internal acoustic meatus. The structure of the receptor organ, that is organ of hearing, sensory organ. You see, this is the osseous spiral lamina. There, this is the basilar membrane, and the tectoral membrane is not shown here. The basilar membranes contain the hair cells and supporting cells. There are inner rod cells, outer rod cells, and the inner hair cells. So they form a conical tunnel. They form a conical tunnel, that is tunnel of Corti, which here lies the cotyledons, which is cotyledons. The structure shall be discussed just now. The organ of Corti is spread like a ribbon along the entire basilar membrane. The thinner inner portion towards the spiral lamina called the zona arcuata, and the thicker outer portion is known as zona pectinate. So zona arcuata and zona pectinate. You see here the thinner portion is called the zona arcuata, and the thicker portion, the dotted line, is the zona pectinate. The cells of the organ of Corti. The cell arrangement at the base membranes are as follows. The two layers of rod cells, one inner and one outer, forming a tunnel of Corti. The inner rod cells are total 3,500 in number, and outer rod cells are about 12,000. The rods are expand like on the top. The rod that touches the top. The outer aspect of these rod cells lies a single layer of inner hair cell, which is also near about 3,500 in number, whereas the outer side of the outer hair cell, outer rod cells, there are 3 to 4 layers of outer hair cells, the sensory cells. So, you see, these are the 3 to 4 hair cells, outer hair cell, this is the inner hair cell, single row. The total number of the hair cells, 3,500 approximately, whereas these are about 13,000, 3 to 4 layers. The hair cells are supported by the pillar cells that means outside there, the goiter cells, the Hensel cells, the Bochet cells, and the cells of Porteus. These are attached on the lateral aspect inside the bony cochlear duct at the spiral ligament. The strata vascularis, these contain vascular epithelium and secretes endolimbs. It is attached to the outer aspect of the cochlear duct. This is the idea for attachment. This is called strata vascularis. You can see another picture also. You see, this is the picture. Here, they are the cells, supporting cells, Deuter cells, the Claudia cells, and Bucher cells. These cells are ultimately they are the spiral ligament attached to the laterally the stria vascular elements are different. So, tectorial membrane. What is the function of this tectorial? It is a gelatinous sheet-like structure which floats in the endolymph in the organ of body. It floats during the movement of the tectorial membrane. During the movement of this tectorial membrane, touches the tip of the outer and inner cell and stimulation, leading to the electrical stimulation. The movement of the tunnel of Corti is a triangular in cross section formed by the rod's outer and inner rod cells and the hair cells. The lying outer aspect of the rod cell, there are hair cell. Lying outer aspect of the outer rods, there are two types of hair cell. The backsepid cell is a type 1, which is richly supplied by the afferent cochlear nerve. Each have a cup-shaped nerve terminals. The type 2 cells, these are tubular in shape and arranged in 3 to 4 rows and receive the efferent in a olivary complex, from the olivary complex. So you can see the cells. The cells, outer here flap-shaped, the calyx almost covering the cell. All this is the calyces of the nerve calyces ends at the tubular cell, type 2 cells. So the one there is projection of the hairs from the cell microcilia. The kinocilia, the smaller one, arranged in a definitive pattern. One is the stereocilia. So stereocilia is the smaller one, is kinocilia. Next. So difference between the outer and inner hair cells. So I have in tabulated manner so that you can understand, you can remember. The shape is flat-shaped, shape is cylindrical. Number, inner hair cell 3,500, whereas the outer hair cells near about 12,000. And number of rows is 1 row, number of 3 to 4 rows. Nerve supply mainly apparent, and the outer hair cells are mainly apparent. Development, they develop early, whereas the outer hair cells develop late. Function, they transmit the auditory stimulus. They modulate the function of the inner hair cells. That means the outer hair cells has function of modulating the inner hair cells. That means sometimes they can control the intensity. The ototoxicity is more resistant in inner hair cell, where the outer hair cells are easily damaged. The high sensitivity is noise. So more resistant to the noise sensitivity, the more noise resistance inner hair cell, where they are damaged easily by the noise sound. So sound, that is sound toxicity and ototoxicity, both of them damage the outer hair cell more. Generation of OAE, autoacoustic emission, is only from the outer hair cell, not from the inner hair cell. The vestibular end organ, that means the end organs of balance. These are the macula of the utricles and sacules and crista of the ampullary portion. These are the vestibular end organs. The macula in the utricle lies in a horizontal plane, where that of the sacule in the vertical plane. That is on the sacule in the vertical plane. The macula of the utricle has approximately 3,000 hair cells, larger than the scalar macula of the utricle, like only 1800 hair cells in the saccular macula. So macular sacula, macula of the utricles having larger in size as well as larger in cell concentration. The stereola is a narrow curved line in the center dividing the macula into two portions, sensory epithelium and the otolic epithelium. So macula of this two, utricle and another macula, have a small narrow curved line at the center, that is not the stereola. The polarity and curvilinear shape of the stereola allows the central nervous system a wide range of neural information at an angle and different angle and three different phase that optimal perception and compensatory correction neuroepithelia of the macula. There are two types of cell I have mentioned, the type 1 cell and type cell similar to the macula, cupula of the utricle. The type 1 cells are having larger concentration in the region of the stereola. There are the surface of each type 1 and type 2 cells projects from the surface to project a numerous stereola, and one thicker one and taller one is the kinocilia. The kinocilia are towards the kinocilia in the utricular macula, whereas the saccular macula, they are away from the stereocilia. That means arrangement of the otolith membrane, it consists of a gelatinous mass that is in a subgelatinous space containing crystals of calcium carbonate known as otoconia or statoconia. The otoconia are multiple small cylindrical or hexagonal particles with pointed ends, consist of calcium carbonates with organic protein matrix, and hence the gelatinous mass, sometimes also called the statoconical membrane, because of this otoconia, particular shape of the otoconia. The ultrastructure of the macula. The crista ampullaris is a specialized neuroepithelium. Crista is the epithelium known as the crista, located in the ampulla. The crista has also hair cells like that. The hair cells of the cochlea also, supporting cells and gelatinous mass or cap-like covering called the cupula. So hair cells are two types. One similar, the class-separate cell with enclosing nerve calyces all over the body, and the type 2 cells cylindrical with no nerve calyces. Type 2 cells are found more towards the periphery of the crista. The hair cells contain one kinocilia which is taller and prominent. So movement of this kinocilia by the fluid movement leads to the depolarization and depolarization depending on the type of direction of the fluid. So this is the ampullary end inside the bony ampulla. This is the crista, the cells arrangement in the crista ampullaris. This is the class-separate cell. This is the rod-separate cell. The rod-separate cells are in the periphery. The class-separate cells is over the center. And these kinocilia are the long projection of the cells. The gelatinous covering and lead to this cupula. The cupula is a gelatinous covering of this solid area. So movement of this fluid here and there leads to the movement of the cupula and the hair cell stimulation, leading to the information you get. The hair cell in the crista are also like this, class-separate, and they are type 2 cells. So this is what I discuss is all about the anatomy of the mid layer. and inner ear, sorry,